Thank you very much, James, and thanks to the organisers for the invitation. It is a privilege to be here. What Theresa and I are going to talk about today is work that is ongoing and has arisen out of CAPREx funding, Alvarado Trust funding, that has allowed us to have leverage to obtain substantial funding from the Leverhulme Trust and Royal Society. So I think that's a real outcome from it. So I've worked for the best part of the last 25 years on African trypanosomes. You might think that that's a very strange thing to work on in Cambridge, but it came about through a combination of really 2 or 3 individual factors. The first was that a few hundred yards from here is one of the only stone buildings in the world that I know with the word parasitology engraved on the front. If anybody knows of another one, I'd like to find out. This was established around 100 years ago for entirely the wrong reasons by Percy Mortino, but over the last 50 years has been a centre for really understanding, trying to understand the molecular details of how parasites cause disease. I first went there just after I'd finished my PhD, which was in botany, and I accepted the job that was kindly offered because at the time the British government had gone through one of its periodic cuts and it was the only job I was offered. So I can't claim there was any career plan for it. And the third thing, I think, was something from my father, who I suppose spent 1957, 1956 and 57 in Ghana as the last part of the imperial administration. And I think he always loved the place, although his views on what should have happened in Ghana are best not repeated here. But he was typical of his generation in Britain. So, why have I kept on working on African trypanosomes, and why is it important? I think this has only come home to me recently. When you first start on research, you think you're enamored of the molecular mechanisms, and it's what I've been trained to do. But when you look at it in detail, what you actually find is that the real problem that African trypanosomes cause, especially today, is not really the human disease. There aren't that many cases. I think in the last 10 years in Ghana, for example, there have been less than 10 cases. It's the cattle disease that's important, and cattle have always been important in Africa. How they got to Africa is still a subject of debate. The most likely explanation is some of them were actually domesticated in northern Africa. And here we see a longhorn cow drawn 6,000 to 8,000 years ago in a part of the Sahara that is now desert, but at the time was agricultural land. These cows and shorthorn cows are related to the European cow, and they've been in Africa for 5,000 or to 10,000 years. They're relatively resistant to trypanosomes as a result of that time. This is the second sort of cow, a zebu. It's got the hump. It's derived from much more recent imports in the last 1,500 to 2,000 years from Southeast Asia. And this cow, this bull is susceptible to trypanosomes. And I took this on a visit to the vet school farm in Achra, funded by CAPREx last year. And this was a magnificent animal, but I had to bend down to take this photo, and I'm not that big. Compared to the agricultural bulls I'd been used to seeing at shows on August bank holidays in Wales, this was a small animal, and part of the breeding to remove the sensitivity to trypanosomes for reasons we don't understand means that the cows and bulls are smaller. Here's its harem, or harem, and you can see that now by standing up I tower over these cows. If you've ever seen a Frisian or a cow on Midsummer Common, you'll know that that's not the normal size that we're used to here. The second thing you'll notice is that the ribs are showing. And I remember at the time Theresa was there, as you'll see in a minute, and this was really good news for us, although not for the cows, because we knew the ribs were showing because they were carrying trypanosomes, and so we'd get some results from the work. This is why, the major reason why ribs show on cattle in Africa. So here we are looking at them. Here's Theresa doing the work, and here's me. I remember at that photo being taken thinking, God, I hope I've put enough insect repellent on. So that was it, and it was a great privilege to go out there, but also in the background, you can see how small the cattle are. So, how does the trypanosome get into the cow, and how is it transmitted from one cow to the other? It's transmitted by this fly, the tsetse fly, which, as you can see from the size of the abdomen, probably is, when it has a blood meal, thinks it's a college feast or something like that. But it will then, the trypanosome cycles through the fly and is reintroduced into another cow 10 days later, really. Now, the distribution, so the trypanosomiasis requires the tsetse fly to be transmitted. And when you then look at the distribution maps of cattle and tsetse flies in Africa, the tsetse flies, or one type of tsetse flies in red, you can see that the cattle in yellowy, orangey, browny, and the tsetse flies really don't overlap so much. This is the major constraint on cattle rearing in large parts of Africa that would be otherwise suitable to it. So, how do the trypanosomes actually make the cows ill? Well, here's a sort of slow motion film of them. These are red blood cells. They grow and proliferate in the blood. And because of this, they're continually exposed to the host immune system. And from early times, this was an interesting phenomenon because the question really was, well, why aren't they killed by the host immune system? Because in fact, if you do certain experiments in a test tube, they are killed. They're relatively readily killed. The answer to this problem has really come from work by lots of people over the last, well, it's really 100 years, and started with this observation on the left. This was a sailor that got back to Liverpool before he became seriously ill. And what this represents is the count of trypanosomes per volume of blood over a time course, in this case of 5 weeks. And what's really unusual about this, and I've sort of drawn it in a fantasy nice curve here, is that it goes up and down by orders of magnitude. And what happens is that in peak A, eventually you'll kill them. But during that time, some of the trypanosomes in peak A have changed and will grow up again as B. Some of these have changed and they'll grow up again as C. And by the time it gets to C, the animal that it's in is capable of killing A and B, but not C. So what happens, it said it's undergone a variation, and this is the antigenic variation. And so this enables it to maintain the infection for, in fact, years. There was a recent case at a London neurological hospital where the patient was believed to have carried the human infective form for 20-something years before the disease flared up. So this can go on for a long time, theoretically. How does it do this? Well, the outside of the trypanosome is covered by the coat of a single protein, and over the years and years, a lot has been found out about this. And what happens is that 10% of its genome is dedicated to making different versions of this protein. So this protein serves to protect the cell from the host immune system and to vary so that the population can survive and maintain an infection. We know an immense amount about the molecular detail of how this works. And don't worry, I'm not going to try and explain this to you. So this is just to illustrate. Some of this is a summary diagram of hundreds of person years' work. But what we don't know is how this works in infections. Occasionally in the laboratory we will use mice. It's not really a good model because these are diseases of large mammals. And over the last few years I've been trying to formulate some questions about this. So for example, what I've just told you about it being able to maintain an infection for years, is that true in a large cow? We don't know. How long can any one trypanosome infect? And it's questions related to this that led to the collaboration with Theresa, who is now going to tell you how we're going to go about and answer the questions, with any luck.
So I'll continue by telling you on what we are doing so far with animal trypanosomiasis in Ghana. So there are various species of trypanosomes that affect both humans and animals, and we are interested in the animal disease. So for example, in West Africa, some of the species that have been detected in livestock are the T. congolense, vivax, and then the T. zoo. And then in some animals, there have been detection of the T. brucei. And in the humans, there are two main species that causes disease, the T. brucei rhodesiense and then T. brucei gambiense. So in Ghana, animal trypanosomiasis prevalence varies widely across the whole country, so from the north to the south of the country. And the prevalence ranges from about 5% in low-risk areas, such as the Dangbe West and also the Nanton district in the north of Ghana, to up to 50%, which usually occurs in high-risk areas such as the Damango or the part of the Volta region that is east to Achra. Currently, there have been several experiments in Ghana that have shown the different species of trypanosomes that can infect cattle in the country. But at the moment, there is no information or there's no data on lifetime infections, which means how the trypanosome can infect a young cattle, for example, over a period of time. So our collaboration is mainly to investigate or characterize trypanosome infection and also follow up lifetime infection of cattle. And with our project, we hope that we'll have a better understanding of the trypanosome biology and also will help to inform control measures in the affected areas in Ghana. So for our project, we had three main aims that we wanted to look at. The first one is that we wanted to know whether individual cattle remain infected by the same isolate or there are waves of different isolates appearing as a continual infection. The second point is to investigate whether there is competition between different trypanosome species, or does one species predominate in mixed infection. So this point means that you have sometimes the infection with two different species of trypanosome, and what happened to these two different species over a period of time. And then we also want to know the correlation between age and the predominantly infecting trypanosome species. So to answer our questions, we decided on two locations in Ghana, one in Achra and the other one in Adidome, which is about 200 kilometers east of Achra. So in Achra, you have some savanna-type vegetation, and in this area you have low tsetse density. Mark have already mentioned that the parasite is transmitted by the tsetse fly. And then it has low prevalence of the disease. And in this location, we decided to choose this location because we wanted to investigate at least two different breeds of the cattle. In Adidome, you have high tsetse fly prevalence and high disease prevalence also. And the type of animals that are available are the West African shorthorn, the zebu, and then crosses from the two animals. So for the project, when we started, I had the opportunity to get the CAPREx fellowship. So last year, in October to December, I was in Mark's lab. And when I was in Mark's lab, what I was supposed to do was to try to start up or do some preliminary work on our main project. And what I was supposed to do, or what I decided to do, was to be able to select or choose primers that will help us to detect or amplify the DNA of the trypanosome in blood of the cattle. So when I was in Mark Lab, I worked on several sets of primers, and I was able to select specific set of primers that will help us to identify the trypanosome. And the aim of this experiment that I did was to be able to differentiate different species of trypanosome depending on the size of a PCR product that you generate after PCR. And on this gel, what I did was I used one set of primer, and I tested it on 6 different trypanosome species. All this DNA was provided by MAC. And what you can see is that with a single PCR, or what's called a nested PCR, you are able to differentiate by the length or the size of the product the different species of trypanosome. What does this mean? It means that, for example, if we go to the field and we take different samples of blood, we should be, and we amplify the DNA, we should be able to differentiate different species of trypanosome just by looking at the length of the PCR product. Then the next question that we asked was, how many parasites are we able to detect at all in the blood of an infected cattle? So for example, if you go to the field and you take, let's say, 10 ml of cattle blood, how many parasites will you be able to detect? So in the lab, what I did with the help of James Wood, we had blood from cattle, and then in the lab, we put some different concentrations of trypanosomes, and then we tried to detect their amount of parasites in this blood. And what I could show was that we could detect at least 1 parasite in 1 ml of blood. So what it translates is that if you go to the field and you take 1 ml of trypanosome of cattle blood, and it's infected with even 1 parasite, you should be able to detect that parasite with our method. So the work that I did in Cambridge, now we have to translate back into the field samples that we collect. So this slide shows actual results that we have obtained from our work. So for our work, what we did was to select 20 animals from each of the locations. And we take blood from each of the animal, and then we follow this animal over a period of time. And for this project, what we want to do is to follow them up to 2 to 3 years, so that we can know the diversity of the parasite and also the dynamics of infection. So here what you see is that with the Achra samples, we could amplify trypanosome DNA from 11 out of the 20 samples that we collected, which amounts to about 55%. And this is a typical result that we also obtained from Adidome, which is at the other site. And what you can see is that here we are able to amplify or detect more trypanosome from more of the samples that we took. And here we could detect about 85% from the samples that we collected. So far we have 4 time points that we have collected so far. And what we are trying to do is to verify some of the data that we have obtained. But the summary of what we have so far is that for most of the cows, the infection is in both of the location. But what we have is that there is higher prevalence or higher infection rate in Adidome relative to the Achra. And then the infecting species are present throughout the time course. So for the 4 time points that we have taken, we could see the trypanosome DNA in almost all the time points. And then there is evidence of reinfection after treatment. So in one of the locations, after the 2nd month, the animals were treated against trypanosomiasis. But in the next consecutive months that we went there, we still saw some infection again. And then we can see that there are evidence of mixed infection. So it means that we could actually detect different species of trypanosome in the same animal. So when I was in Cambridge, apart from the science, I also had the opportunity to do other things. So Professor Carrington, and during my preparatory work, suggested that I visited this lady, Dr. Kim Pigosi. She is an expert in animal trypanosomiasis, and she has worked with a lot of scientists in Africa. And I think that the meeting that I had with her was very useful because she suggested very, very useful primers that I could use to improve on my methodology. And while I was here also, I had an opportunity to meet with other CAPREx fellows, and we had opportunities to not to only attend presentations, but also to go out and have some fun. And this is some of the people that I met with Jenny and everyone that was involved. Also, I had an opportunity to attend some very, very important seminars in Cambridge. So the first one was the CAPREx Research Showcase, where we had the opportunity to present our work to a bigger public in Cambridge. And also, I had an opportunity to be invited to a roundtable discussion. And also, I attended some of the King's College seminars, which was very interesting for me. So how far have we come with our collaboration? I think that this collaboration has been very, very great because apart from the CAPREx fellowship, we had an opportunity to get an award by the Liverhome Royal Society Africa Award, which Mark have already mentioned, and we have to continue, or we're going to work for a period of 3 years. Now, back home in University of Ghana, I have a fully functional lab that I can work in. So when I collect the sample, I don't have to bring it to Cambridge, but I can work on all the DNA extraction and the PCR and everything back in Ghana. Through our collaboration under the Royal Society Africa Award, Professor Carrington and I have a PhD student who is on the project. So basically, we have co-supervision of a PhD student, and the PhD student started last year, and in Ghana it takes 4 years, so about 3 years more for her to finish her PhD. And in July this year, Professor Carrington and I also organized a workshop, which was the workshop on molecular biology, pathogenesis, and diagnostics of neglected disease. In this workshop, we had students from about 5 different institutions in Ghana coming to University of Ghana to learn some diagnostic methods that they can apply back in their own universities. And then currently what we are doing is trying to collect more samples to continue on our project. So I would like to say a big thank you to Professor Carrington for the opportunity to collaborate with him, and also the CAPREx Fellowship for the opportunity, and also the Aborada for money, the initial money to buy some equipment, and also my other colleagues back at University of Ghana. Thank you very much.